Hello everyone, welcome back to the class. In our previous class, we discussed about the structure of eukaryotic chromosome. Let's revise what we learned yesterday. The difference between eukaryotic chromatin and chromosome. Chromatin is organized structure of DNA and protein found in nucleus of eukaryotic cell, and chromosome is highly condensed. Chromatin is less condensed. Chromatin is extended DNA. Chromosome is packed DNA. Chroma means color, some means body, strongly stained by particular dyes. In interface nuclei, chromatin plus nuclear matrix will be formed. DNA sequence is attached to the nuclear matrix at the matrix attachment region, that is MAR. This is the structure which shows the chromosome compactly arranged, and this is the structure of chromatin, which is extended with a protein. DNA is wound around the protein. We also discussed about the chromatin and its different subtypes, that is euchromatin and heterochromatin. Euchromatin is light staining, less condensed portion. Heterochromatin is dark staining, highly condensed portion. So light means it is transcriptionally active, dark means it is transcriptionally silent. And also we discussed about the subtypes of heterochromatin, that is constitutive and facultative. Constitutive remain condensed throughout the cell cycle and development. Facultative, where the region of euchromatin can be converted to the heterochromatin and vice versa. So here in constitutive heterochromatin, they are transcriptionally inactive. Here they sometimes has the potential for gene expression. We also discussed about the chromatin DNA molecule has three specific nucleotide sequence, that is centromere, telomere, and the origin of replication. Centromere is constricted region of the eukaryotic chromosome, where you can see the sister chromatids or the chromosome arm is attached, and there is a presence of kinetochore, which is a complex protein which during the mitosis helps in segregation of sister chromatids by binding the spindle fiber to it. There are 4 types of centromere based on the location of the centromere. There are 4 types of chromosomes based on the location of centromere: metacentric, submetacentric, acrocentric, and telocentric. And we also discussed about the telomere, which is specialized structure which caps the end of eukaryotic chromosome, and the special unusual property of telomeric sequences as it has a presence of G-rich single-stranded 3' overhang, and also there are some 2 sequence specific DNA binding proteins: telomeric repeat binding factor TRF1 and telomeric repeat binding factor 2 TRF2, which bind directly with telomeric sequence, which in turn interact with the larger proteins. And also we discussed about the origin of replication, where it is the region or the site where where the DNA starts replicating. Eukaryotic chromosome is having a feature of multi-repliconics. They have multiple origin or ori site, and it is very helpful in DNA replication. So today we'll be learning about chromosomal organization. What is this chromosomal organization means? So we in G0 phase, or in the initial phases of cell before dividing, dividing, the cells will be having less condensed, loose, extended strand of DNA known as chromatin. So in G0 phase or in interphase, we can see a thread-like structure which is not compact, which is free, which is loose. When we see the chromosome in the M phase, so it is somewhat compactly arranged. In anaphase, it still binds, or it binds to further proteins, and it forms a compact structure, darkly condensed. So it forms a particular compact structure when there is a last phase of the cell division. So there are several organization of chromosomes. So this can be said as first level, this can be said as second level, this can be said as third level. So there are particular levels of organization. Why this organization happens? It aids in proper segregation of chromosome or the genetic material while cell is undergoing division. And this extended structure has a disadvantage. Some of the genetic material or the sequence can be lost during the process of cell division. So it is important to pack, it is important to bind, so it forms a complete, or it forms a stable molecule, it forms a stable structure for further processing. So that is why the chromosomal organization happens. In G0 phase and interphase, DNA exists as a nucleoprotein complex, that is chromatin. In M phase, it is converted into more condensed form, that is chromosome. In anaphase, single enormously long linear DNA molecule plus protein that fold and pack the fine DNA thread into more compact structure. So the protein which binds to the DNA or the binds to the chromatin for which helping the packaging of DNA are of two types, that is histone chromosome protein and non-histone chromosome protein. So these histone chromosome proteins are basic proteins and they are synthesized during S phase. Non-histone chromosomal proteins are acidic in nature, they are synthesized throughout the cell cycle, example topoisomerase. Then in histone chromosomal proteins we come across two subtypes, that is replication dependent histone, example H1, H2A, H2B, H3 and H4. Replication independent histones, example H3.3. So there is a word known as histone, right? Histone plays a major important role for organization of chromosome of the first level. So there is histone, remember this sequence, histone helps binding of DNA or chromatin. So histone helps in binding of DNA or chromatin network. Histone is nothing but a protein. So here DNA plus protein forms the first level of chromosomal organization. So the DNA plus protein is known to be nucleosome. This is C, nucleosome. So DNA plus protein, histone protein, forms a structure called nucleosome. This nucleosome further helps in chromosomal organization of second level, third level and fourth level. So let us now discuss about histone. Histone are responsible for first and most basic level of chromosome organization, the nucleosome, a basic structural unit of chromatin. Nucleosome contains histone octamer, that is 2 copies of H2A, H2B, H3 and H4. H3, H4 tetramer are flanked by 2 H2A, H2B dimer, which is a core histones. They are rich in lysine and arginine, positive charged amino acids which can effectively neutralize the negatively charged DNA backbone. These 8 proteins form a barrel-shaped core octamer with the DNA wound around the outside. So to explain the histone protein structure or the nucleosome structure, the nucleosome, as discussed earlier, the nucleosome is made up of DNA and histone proteins. I write it as HP, histone proteins. So the organization, there is a particular organization of nucleosome. Nucleosome is considered as histone octamer. Octa basically means 8. So there are 8 histone proteins. Which are they? So histone is the 8 proteins form a barrel shape. You know barrel? It will be like this. barrel shaped. So the structure of the nucleosome is somewhat similar to this, barrel shaped. So the octamer is having 8 proteins. Which are they? They are H2A, H2A, H2B, H2B, H3, H3, H4 and H4. Count 1, 2, 3, 4, 5, 6, 7, 8. So this H2A, H2B are dimers. So there are 2 H2A, 2 H2B, 2 H3 and 2 H4. So all these form a octamer. How do they arrange? So this is a schematic representation. H2A, H2A, H2B, H2B, H3, H3, H4 and H4. To label this, H2A, H2A, H2B, H2B, H3, H4, I'll write it like this, H3 and H4. So this is a nucleosome barrel. The 8 proteins form a barrel shaped core octamer with a DNA wound. So as I said, it is a barrel shaped. The nucleosome is barrel shaped, to which the DNA wounds. To which the DNA wounds. See here, nucleosome provide the first level of DNA packing. So this orange one is a histone, protruding histone tail. This is a protruding histone tail, but this is a nucleosome core, which it is having 2 H2A, 2 H2B, 2 H3, 2 H4. Okay, to which the blue color strand is DNA to which it is bound. The nucleosome surrounded by the DNA. It gives a basic beads on string. See here, beads on string. On treatment with micrococcal nuclease. Micrococcal nuclease helps in breaking down and leading to formation of mononucleosome. And in addition to core histones, there is another histone protein, that is H1, which is considered as linker DNA. What is its function? So it links the DNA, linker of DNA. It is known as linker histone. It links the DNA and it is associated with linker DNA and may lie at the point where DNA leaves the nucleosome. See, in this structure you can see the green color part, which is a histone H1. So it is connected with, it is lying at the point where DNA enters and and leaves the nucleosome. Single linker histone is attached to each nucleosome chromosome. Then how this nucleosome assembles? In a nucleosome assembly, histone binding to DNA is thought to occur in two-step process. So a tetramer or 2-dimer of histone H3 and H4 first deposits on DNA. Then 2-dimer of histones H2A and H2B are deposited to yield nucleosome core. Which is coming first? H3 and H4 dimer first deposits on DNA. After that, H2A and H2B deposits to yield the nucleosome core. Nucleosome assembly proteins NAP helps in, or it is termed as histone chaperones, helps in incorporation of histones into the nucleosome. So these histone proteins are present in the cytoplasm. So the nucleosome assembly protein, which are termed as histone chaperones, help in entry of histones into the nucleosome, so that the histone will help the organization. So histones are closely escorted by histone chaperones from their point of synthesis up to their delivery site. Many histone chaperones are identified: nucleoplasmin, ASF, anti-silencing function, HIR, histone regulator, CAF, chromatin assembly factor. So this factor helps in attachment or assembly of chromatin. NAP1, that is nucleosome assembly protein. That protein helps to deliver and helps to assemble the nucleosome. Then this is the second level of chromosomal organization. So it is a second level of organization. So till now we know that nucleosome has been come to the picture, where the chromatin network is wound, or the DNA is wound to the nucleosome, forming a beads on the string type of structure. This is the first, and it is having 11 nanometer. Remember this, it is having 11 nanometer diameter. Okay, then comes the 30 nanometer fiber, where this component arranges in two different styles. Based on the arrangement of nucleosome, or based on the in vitro data, there are two models which is suggesting the fiber, that is solenoid model and zigzag model. Which are those? Solenoid model and zigzag model. To explain solenoid, it is just a one start helix. It is having one strand, we can say. Nucleosome follow each other along the same helical path. This solenoid is a single start. The helix which I have drawn is nothing but a DNA, and there is a nucleosome attached to it, having a bead structure. So which I draw previously was a linear one, and this linear one with the nucleosome has form a helical path. There is interaction between nucleosome which occurs sequentially. It is characterized by interaction between consecutive nucleus, n plus 1. This is n plus 1, then n plus 2 and n plus 3. So there is interaction between the histone core. This is a solenoid. Okay, solenoid. Another one model is there, that is zigzag model. Zigzag model is two start or two strand helix. So here there are two strands. Strand 1 and strand 2, which are having nucleosome, where the chromatin is wound, or the DNA is wound around that nucleosome. So this forms a zigzag structure. There are two strands. Okay, strand 1 and strand 2. They form zigzag structure. There are nucleosome here and there. Okay, there is a interaction between them. That is the interaction is n plus 2 and so on. You'll get to know this difference between solenoid model and zigzag model with the help of image. See, it is one start helix model. Zigzag model is two strand helix model. The nucleosome follow each other along the same helical path. So there is interaction between the consecutive nucleosome, that is n plus 1, n plus 2. Here the two strands containing stacked nucleosome are wound into a left-handed two strand helix. So in each strand, nucleosome are aligned with each other like a stacked coin. When you stack a coin one upon the other, so that structure will resemble that nucleosome structure is similar to that structure. So interaction between alternate nucleosome, that is n, n plus 2, n plus 4 and so on. See here, this is the solenoid model. Solenoid model is a single and it forms a helical path. Zigzag is a two-strand structure which forms a stacked nucleosome. So all these images are the solenoid model itself. It is a one start. One start is classical. This is cross linker, random fiber, zigzag ribbon. So all these are solenoid model itself. Okay, this was about the nucleosome structure of 30 nanometer fiber. Now is metaphase chromosome. In interphase, there was a 30 nanometer fiber which is a major type of chromatin in nucleus. So when nucleus divides, DNA adopts more compact form of packaging. So till now what all we discussed? So we discussed first organization where the chromatin or the DNA is wound to the nucleosome, which was the first level. Second level where these beads on string will form a helical or solenoid model or what zigzag model, where the nucleosomes are found here and there is interaction between them, and this is a second level. This was how much nanometer? That is, how much nanometer was it? So first was beads on structure is 10 nanometer and this was 30 nanometer. Now we are studying about the third level, that is metaphase. In metaphase chromosome, what happens? So there is a protein scaffolding happening, where it helps to form a compact packaged structure. So that is metaphase chromosome. So DNA is very compact and the loops of 30 nanometer chromatin fiber containing 20 to 100 kb of DNA loop are attached to a central scaffold. So there is a protein which gives a support. There is a protein. So there is a protein to which these loops, the loops of chromatin, are attached to the central scaffold. Here also there are the loops. Okay, so that forms a compact structure. This scaffold consists of many proteins, that is non-histone acidic proteins, non-histone acidic proteins, and there are chromatin proteins, and there are condensin proteins. Non-histone acidic proteins, especially topoisomerase 2. The function of topoisomerase 2 is it is an enzyme having ability to pass one DNA double helix through another by cutting and sealing both strands. So this topoisomerase 2 is an enzyme which cuts the strand and it seals the strand of different DNA double helix. Okay, chromatin proteins, on the other hand, it binds to AT rich sequence chromatin loops attached by stretched of several hundred base pairs of highly AT. So the DNA where it attaches to the scaffold, that region is known as scaffold attachment region, where this chromatin proteins forms a loop. That chromatin loops are attached by stretched of several hundred base pairs of highly AT rich DNA. Then condensin protein, it maintains the condensed structure of chromosome. They are made up of SMS, that is structural maintenance of chromosome proteins. And last one, that is Cohesin, it links together the sister chromatids immediately after replication and keeps them together. To revise, let us see the details. See, the scaffold consists of several proteins, that is topoisomerase 2, which is a non-histone acidic proteins, chromatin proteins and condensation proteins. Topoisomerase 2, the function is, it is having the ability to pass one DNA double helix through another by cutting and sealing both strands of DNA. The chromatin proteins, which binds to AT-rich sequence of chromatin loop attached by the structure of several hundred base pairs of highly AT-rich DNA, that is scaffold attachment region. Condensation protein maintains condensed state of chromosome, and it is made up of SMS protein, that is structural maintenance of chromosome protein. Then cohesin, it links together the sister chromatids immediately after replication and keeps them together. This cohesin has its function during anaphase and during metaphase also, where the sister chromatids is attached to the centromere by the help of this protein itself. Condensation makes chromosome highly condensed. So in next picture you will come across the clear picture of chromosome packaging. See here, the chromosome scaffold components, the proteins which we discussed right now, that protein is acting as a support system for these loops, and the coiled chromonema fiber, which is a chromatid at metaphase, becomes more compact to form a compact structure. Okay, the diameter you can see from 32 it has increased to 100, and it can also extend up to 300 nanometer. So the 300 nanometer chromonema fiber are found in later stages. In prophase it is 100 nanometer chromonema fiber. Okay, and there is a loop chromatin here, which has extended outwards. So from here, from nucleosome model, or from beads on string to the compact metaphasic chromosome, it is nothing but the structural organization and the process of making chromosome compact. See here, this image depicts all the sequence of the organization. See, at the simplest level is the double-stranded helical structure, which is having DNA double helix, 2 strands, complementary base pairing, which is of 2 nanometer. In second level, DNA is complexed with histone to form nucleosome. So you can see it is becoming compact. DNA is complexed with histone. This yellow color patch you can see, that is histone H1, which is a linker histone. Each nucleosome consists of 8 histone proteins around which the DNA wraps. So the histone proteins are basic in nature, and they are positively charged. DNA is negatively charged, so therefore they help in neutralization, and therefore they help in attracting them. Then nucleosome forms a beads on DNA string, which is of 11 nanometer in diameter. Then the nucleosome fold up to produce a 30 nanometer fiber based on the arrangement of the nucleosome and the strand, which is the start strand, that is single strand start or double strand start. There are 2 models, solenoid or zigzag model. In next, that forms a loop averaging about 300 nanometer in length. At last, the 300 nanometer fiber are compressed and folded to produce 250 nanometer wide fiber. And at last, there is tight coiling of the 250 nanometer fiber produces the chromatid of a chromosome, which can say the chromosome is compactly arranged. In our next class, we'll be learning about histone modification. So in this, there are several topics like acetylation, methylation, phosphorylation, sumoylation, ubiquitylation, and ADP ribosylation. So there are many functions which are characterized by each of these processes. So that will be discussed in the next class. Thank you.